now being transcribed.
Thank you once again. Thank you, sir. Thank you, Principal Sir, for inviting the guests. Before we proceed to the speaker introduction, I would like to share few words about my today's speaker. So our today's speaker is my guide, PhD guide, Jubie Madam. I'm very fortunate to pursue PhD under the guidance of Madam. I have learned so many things from the Madam during my PhD regarding research and how to plan a research and how to execute that reactions, everything. Madam is a very good researcher and dedicated and hard worker. I'm always proud to be a student of the my guide, Jubie Madam. So, Madam received many best researcher awards from JSS University and Tamil Nadu State and from APTI. The students who did the PhD under the guidance of the Jubie Madam are in very good positions across the world. Thank you, Madam. I am very thankful to you throughout my life. Thank you, Madam, for accepting our invitation in the your busy schedule. Now, I would like to request Anusha Madam, please introduce the our today's speaker, Dr. Jubie Madam, Associate Professor, Department of Pharmaceutical Chemistry, JSS College of Pharmacy.
Good morning, everyone. It gives me immense pleasure to welcome you all to today's session of our national level 1 week e-FD on emerging research trends in pharmaceutical sciences. We are honored to have with us a highly accomplished and inspiring academician, Dr. S. Jubie Ma'am, Associate Professor in the Department of Pharmaceutical Chemistry at JSS College of Pharmacy, Ooty, under JSS Academy of Higher Educational and Research, Mysore. Dr. Jubie Ma'am brings with her over 23 years of rich experience in pharmaceutical education and research. She completed her B Pharm from SB College of Pharmacy, followed by her M Pharm from KM College of Pharmacy, where she was recognized as the best outgoing student. Ma'am earned her PhD from JSS Academy in 2014. Her major research interests include the discovery of novel chemical entities for cancer therapy using in silico approaches and drug repurposing. Her scholarly work speaks volumes, over 129 publications, a cumulative impact factor of 177.85, and more than 2,000 citations. She has received 7 granted patents, authored 5 books and 8 book chapters, and secured multiple prestigious research grants, including from AICTE, RPS, ICMR, DST, and even international funding from Shwara University, Saudi Arabia. Dr. Jubie Ma'am is also an excellent academic leader, serving as the academic coordinator and actively contributing to quality initiatives such as NAC, NBA, and NIRF. She has successfully guided numerous B Pharm, M Pharm, and PhD scholars and is a frequent speaker at both national and international platforms. Her contributions have been recognized with her several awards, including Pharmacy Women of the Year 2023, APTI Central, Best Teacher Award in 2022, Research Excellence Award 2025, and many more. Today, Dr. Jubie Ma'am will be delivering a session on a highly relevant and timely topic in drug discovery, leveraging in silico drug repurposing for advancements in drug discovery. Her passion for research and education is truly inspiring, and we are privileged to have with her today. Please join me in warmly welcoming Dr. S. Jubie Ma'am.
Yeah, thank you, madam, for the nice introduction.
Thank you, ma'am. It's my pleasure.
Yeah. Yeah, is my screen visible to all of you?
Madam, it is visible.
Yeah, and is it moving?
Yes, madam, it is moving.
Yeah, okay. Yeah. So, a very good morning to one and all who gathered virtually. So, at the outset, I thank the management of Vasantidevi Patil Institute of Pharmacy, and special mention to Dr. Shyam Sundar, my student, for giving me an opportunity to share my thoughts on these in silico drug repurposing, which is an emerging technology in the advancement of drug discovery. Okay, so lighter way we can say an old wine in new bottle. So in this presentation, I'll be covering an overview. What is drug repurposing and what are all the different approaches? A special mention to what are all the in silico approaches we can use it for drug repurposing, and advantages of in silico and drug repurposing in general, and challenges. And finally, I'll be ending up with one case study where we have incorporated 2 to 3 drug repurposing, that is in silico repurposing techniques, and in vitro repurposing also. Yeah. So it all starts with a novelist, a pharmacologist, Sir James W. Black. According to his words, the most fruitful basis of the discovery of a new drug is to start with an old drug. So here, all about the repurposing of existing drugs. Okay, so the drug is originally indicated for a therapeutic purpose. So based on that structure of the drug or mechanism of the drug or transcriptomic structure of a drug, it will be intended for a new therapeutic effect or new pharmacological effect. Okay, so that's all starts from this old drug rather than discovering small molecules. Okay, so why this drug repurposing? So normally, as we all know very clear, the drug discovery, it is a very complex process and it involves around 15 years and $2 to $3 billion of money is involved. It starts with discover and development, okay, and preclinical research where the discovered molecules are tested for its activity at animals. Okay, then it goes for clinical research. We have 4 phases of the clinical trials, and it goes for FDA review, and finally after once approval also it goes for FDA post-marketing surveillance. Now if you go for this drug repurposing, because we are taking a drug which is already proven for its safety, okay, so some amount of money and some amount of time can be reduced rather than starting with the small molecule discovery. Okay, so the years will be reduced, around 4 to 5 years will be reduced. Money is also reduced. I will tell you during my advantages of drug repurposing slides. Okay. Yeah, so this is the proper definition of drug repurposing. The drug repurposing, it may also called as drug repositioning, reprofiling, or retasking. So it's all about the strategy for identifying new uses for approved or investigational drugs that are outside the scope of the original medical indication. Okay, so the drug, it is intended for one therapeutic use. Okay, so it has been approved for that. Now the same drug, it has been used for other indication outside the scope of the original indication. So that is called as drug repurposing or repositioning. So it all based on, the drug repurposing is based on polypharmacology, a field of study related to design, discovery, synthesis, and use of pharmaceutical agents, that is drug, small molecules, antibodies, stabilized peptides, natural products, etc., that act on multiple targets. Okay, so here not focusing on one drug, one target, rather than one drug, multiple targets. Okay, so the genes or proteins which are associated in a particular pathway, that is multiple targets. So this drug repurposing, the origin starts with this polypharmacology. Yeah. So as I told you, like complex disease like if you take cancer or any neurodegenerative disorder or rheumatoid arthritis, so it involves so many pathways. It is not confined to a single pathway. Okay, so if you take rheumatoid arthritis, it involves so many pathways. Okay, starting with North signaling pathway, SYK, P13, JAK, and WNT, MAPK signaling pathway. So it involves multiple genes and multiple proteins, multiple targets. Okay, so these pave the way for this drug repurposing. Now coming to the advantages of drug repurposing. So the risk of failure is low because the repurposed drug has already been found to be sufficiently safe in preclinical models because it has been tested for its safety profile, okay, during its clinical trial. So that, what I can say, that risk of failure will be very less, okay, because we are not completely taking a new molecule. Okay, rather than a new molecule, we are taking a drug which is already proven for its safety. And the second one is the time frame is less compared to the conventional drug discovery. Okay, because most of the preclinical testing, safety assessment, and formulation development will already have been completed. So some physicochemical parameterization also we can take it from the previously approved or previously proven studies. And less investment is needed. As I told you, for a drug discovery, it takes around $2 to $3 billion. But when you go for this repurposing, the cost is substantially, it is reducing. It is coming around $300 million in an average. And even though the regulatory and phase 3 costs may remain more or less same for the repurposed drug for a new drug in the same indication, but we can save the amount, especially preclinical phase 1 and phase 2. And very importantly, when we do this repurposing, we can come to know, or this repurposing, it may reveal new targets and pathways, okay, unexplored targets and unexplored pathways further exploited. Okay. Yeah, so the very good example is this sildenafil citrate. Okay, so we know very well this sildenafil citrate, it is a well-known antihypertensive. Okay, so it is antihypertensive drug, but the antihypertensive drug, when the drug has been used for its therapy, one side effect, okay, it was observed that the sildenafil, it has been used to treat the male erectile dysfunction. Okay, so it is now used as Viagra, and it is a blockbuster drug. The Pfizer's company, it is launching this drug. So what we are trying to observe here is the original intention of this sildenafil is antihypertensive drug. It's one of its off-target effect. Okay, so here the off-target effect is very, very important. The on-target is it's to reduce the hypertension, but it's one of its off-target effect, that is the treatment of male erectile dysfunction. So that off-target effect, now it made this sildenafil to treat this EDF rather than its original antihypertensive effect. So this is the way this repurposing has come to picture. Another very good example is, we know very well, this thalidomide. Okay, it is a non-barbiturate sedative. Okay, so this thalidomide, it is originally marketed in some countries in 1957. Okay, so it is mainly used to treat the morning sickness for the pregnant woman, especially for the first trimester. Okay, but the mothers who have taken this drug, no, okay, so they got this skeletal birth defects. So it was observed in 1957. Later on, the drug has been withdrawn from this therapy. Okay, so it is not at all used for that. But accidentally, okay, it has been found out in 1964 for the treatment of erythema nodosum leprosum, that is ENL. It is a disease which is related to leprosy. It is related to immunological disorders. Okay, and later on, now it is used for the treatment of multiple myeloma. Okay, so now the original on-target effect is it is sedative effect, but it has been used for the treatment. The off-target effect is nodosum leprosum, and now it is used for the treatment of multiple myeloma. Okay, so this is thalidomide, and the what I can say, the derivative of this thalidomide is lenalidomide. Okay, so this is the one more example, very good example for repurposing. Likewise, the stories goes on. Okay, you take zidovudin. The original indication is it was used for cancer, but now it is used for the treatment of HIV virus, that is AIDS. Okay, likewise, minoxidil, another very good example. Okay, it is on target effect is hypertension, but now it is used for the treatment of hair loss. Another one is sildenafil. Okay, then celecoxib. Celecoxib is a COX2 inhibitor. It is a NSAID used for the treatment of pain and inflammation, but now it is used for the treatment of familial adenomatous polyps. Likewise, you have so many drugs. Okay, relaxifen, now it is used for osteoporosis. Now it is intended for breast cancer. Likewise, you have so many drugs which are used for repurposed. Okay, aspirin, analgesia, now it is used for colorectal cancer also. You even in fenfluramine and remdesivir. We know very well this remdesivir, which was used during the pandemic time, COVID-19 in infants and children. So these are the successful stories of drug repurposing. Now, very importantly, the steps. What are all the various steps involved in this drug repurposing? It involves 3 steps. Okay, so the first step is very critical: identification of a candidate molecule for a given indication. So here, the hypothesis generation. Okay, so just like that, we cannot take a molecule and we cannot repurpose for some other indication. So we have to have a proper justification. We have to generate the hypothesis. Okay, so here for this hypothesis generation, and after here we are utilizing the modern approaches. Okay, all the computational techniques, everything we are just justifying the generated hypothesis. That is the first and foremost step for any technique if you use this drug repurposing. Once you identify, okay, this drug has been used for this so and so use. Okay, you have to you have proven by using some in silico techniques. Okay, you have some assumption, you have some prediction, then straight away you have to go the mechanistic assessment of the drug effect in preclinical model. That is step 2. And step 3 is evaluation of efficacy in phase 2 clinical trials. We are skipping the phase 1, assuming there is sufficient safety data from phase 1 studies undertaken as part of the original indication. So these are the 3 steps will be used in drug repurposing. Now, coming to the types of drug repurposing, you can broadly categorize into 2 types. One is in silico repurposing, where you can use different in silico techniques. The second one is activity-based in silico repurposing or activity-based drug repurposing. Okay, so one very basic example is by the structure-based molecular docking. How you can utilize this drug in silico drug repurposing. Okay, So if you see the protein data bank, okay, it has got crystallographic protein structures of so many proteins. Okay. On the other hand, I have the drug bank, that is the database which has got so many drugs or so many small molecules. Okay. So I can pick up any particular protein from this protein data bank. The same time, I can take a drug from this drug bank and simply I can do the molecular docking study, a structure-based in silico study. So by doing so, I can inspect the docked poses. I can find out the potential compounds. Okay. Once I find this molecular docking, followed by that, I can further confirm the molecular docking studies with molecular dynamics, molecular simulation, and finally, I can find out the hit compound. This is one way of in silico drug repurposing. Okay. On the other hand, no. Rather than doing this directly, I can take for any in vitro studies, okay, cell line studies or flow cytometry like cell cycle kinetic studies or gene expression studies like that, or in vivo studies. So these are the activity based study, but here we need the actual drugs. Okay, the physical structure should be there in our hand to do the screening, where in silico drug repurposing we have only the structures of the drugs. Okay, so here these in silico repurposing utilizes public databases and various bioinformatics tools to systematically identify interaction networks between drugs and protein targets. Okay, so one such network pharmacology network only I am going to show in my case study, where we have used the in silico studies. Yeah. Various approaches in silico repurposing. Again, first we can broadly classify into in silico repurposing and activity based repurposing. Again, this in silico repurposing you can categorize into 6 subtypes, starting with signature matching. Okay, where the transcriptomic, the gene expression or chemical structures or adverse structures of drug and disease can be compared. So that is called a signature matching. Here the signature, it may be transcriptomic signature or chemical signature or the adverse effect signature. So that can be matched with disease and disease or drug and disease. Then the well-known structure-based in silico technique, molecular docking study. Then this is genomic GWAS study, genomic variant. Okay, the mutant, where you can identify a single mutant which is very predominant in the disease, and you can compare that mutant with various drugs. Okay, so that is genomic genetic association and pathway mapping. So this is network analysis. Okay, so once you identify the target, so that target can be mapped with variety of drugs. Okay, we can call it as protein drug interaction network. Okay, PDI network. And finally, retrospective clinical analysis, where you can go for this electro EHR, that is electronic health records, and novel data sources. Okay, and the down one are the actual repurposing experimental approaches. One is the binding assays by using affinity chromatography or mass spectrometry, and another one is phenotype screening, high throughput phenotypic screening. So these are the various types of in silico and experimental techniques. Yeah, now quickly we will be moving through one by one. What is what? So the first one is signature matching. As I told you earlier, the signature matching is based on the comparison of unique characteristics or signature of a drug, okay, against another drug, that of another drug or disease or clinical phenotype. Okay, so you can compare with any drug or disease or drug or drug. Okay, so there are 3 general types of data. One is transcriptomic data. The second one is chemical structure. The third one is adverse event profiles. Okay, so these can be compared with disease, drug, or disease or disease. Okay, So let us see the example. The first one is drug-disease similarity. So the transcriptomic signature of a particular drug is derived by comparing the gene expression profile. Okay, so the gene expression profile of a cell or tissue before and after treatment of the drug is observed. The resultant differential gene expression signature expression is compared with the disease-associated profile that has been similarly obtained through differential expression analysis of disease versus healthy conditions. One such example is topiramate. Actually, the topiramate is it is a GABA agonistic activity. It is used as an anti-epileptic drug. Okay. Now it is used for the treatment of inflammatory bowel disorder. How they have observed it actually? The first, they have taken the gene expression profiles of this inflammatory bowel disorder by using, there is a database called NCBI gene expression omnibus. Okay, so it will give you the gene expression signature of the particular disease. Here, the disease is inflammatory bowel disorder. The same thing, there is one more tool which is called as connectivity map. This connectivity map is giving the gene expression profile of 164 commercially available drugs. Now, when they compare the gene expression pattern of disease and drug, the topiramate is very much suited, very much matched with the gene expression profile of the inflammatory bowel disease. Okay. Then later on, of course, they have gone for the preclinical and clinical studies. So this is drug-disease similarity. Similar way, drug-drug similarity. So here, the scientists, they have observed the shared mechanism of action. Okay. Of immaterial, whether they have same structure or different structure, but if they share the mechanism of action, okay, the mechanism of action is same for both the drugs. No, it is observed that they might have got the signature also same, and they will be having therapeutic activity also same. Okay, one such example is Fasudil. Okay, so this Fasudil, it is used for cerebral vasospasm following subarachnoid hemorrhage. Okay, so here the Fasudil, it inhibits a very good kinase, that is ROC kinase, that is ROC inhibitor. Okay, it inhibits the ROC. The same ROC kinase is also involved in a neurological disorder, which is called as ALS, amyotrophic lateral sclerosis. It is a progressive neurodegenerative disease. So here, both the diseases, the ROC kinase is involved. So it is understood that the drug is used for the treatment of ALS. So this is one such example for drug similarity. The second well-known used technique is computational molecular docking technique. So it is a well-known structure-based computational strategy. So here we find out the binding affinities and we predict the binding site, okay, between the ligand and the target. So normally ligands will be small molecules, it may be the drug, and it will, we will find out the efficacy against the target. Okay, if there is prior knowledge of a receptor target involved in a disease, then multiple drugs could be interrogated against the particular target. That is conventional docking. What we do is, we take a particular target and against the particular target, what are all the possible ligands. Okay, so that we find out doing this by doing this molecular docking. So here in this drug repurposing, what we do, we have a lot of drug libraries. So that from that library, I wanted to explore a particular drug. Only this drug I wanted to explore. So that drug I will be taking and I will be docking against the array of target receptors. Okay. It may be a 10 or 15 receptor, I will be docking it, and out of these 10 or 15, which target is giving a very good score and very good binding affinity, 2-dimensional interactions, 3-dimensional interactions, so that can be taken for further repurposing. So one such example is mebendazole. So mebendazole, it's a very good anthelmintic drug, okay, azole derivative, benzimidazole derivative. So now it is used for the treatment of various cancer. It is used for a mediator of angiogenesis, that is vasculoendothelial growth factor 2 inhibitor. It is a mediator of angiogenesis. The third one is genome-wide association studies, that is GWAS studies. So here the genetic variants are identified. Okay, so the GWAS aim to identify genetic variants associated with common diseases and thereby provide insights into the biology of diseases. Okay, so here the particular variant or mutant is identified. Okay, so that mutant is associated with any other disease, so that also checked with the in silico tools. Okay, so this is called as GWAS studies. Okay, so here the example is denosumab. It is used for the treatment of postmenopausal women with high risk fracture of osteoporosis. It targets the TNF-SF11, that is tumor necrosis factor superfamily member, which is called as Rankel factor. So this Rankel factor, and here one such variant is RS2062305. So this particular variant is also used in Crohn's disease also. So now this denosumab, the variant, since the variant is also involved in these diseases, it is repurposed for this Crohn's disease also. So this is the genome-wide association studies. And the fourth one is the pathway or network mapping. This you can explore very well this area. So the pathway-based or network-based approaches have been widely used to identify drugs or drug targets that may have potential in repurposing. Okay, so the network analysis involves constructing drug or disease network. There are two types of networks. You can construct PPI network, protein-protein interaction network, or PDI network, protein-drug interaction network, okay, based on gene expression patterns, disease pathology, protein interactions, or GWAS data. Okay, so example is pranlukast. It is a leukotriene receptor 1 antagonist, a well-known drug for the treatment of asthma. Now it is repurposed for the treatment of Alzheimer's disease. Okay, the second one is amrinone. It is a phosphodiesterase, I think 4 inhibitor, which is used in congestive cardiac failure. Now it is used to treat the viral infections. So what is the basic over here? The genes which are involved in the particular viral disease, that have been taken, and the drug has been networked with that genes which are associated with the particular disease. And finally, based on some parameters, some topological parameters, like how we tell the scores for docking studies, G score or autodoc score. Likewise, we have some parameters. Based on the parameters and the output of that network, you can identify the drugs. And EHRs, okay, so the retrospective clinical analysis, use of electronic health records. So these retrospective clinical data can be obtained from various sources, including EHRs, post-marketing surveillance data, and clinical trial data. So like how we are taking the chemical structure, chemical transcriptomic signature, mechanism of action, similar way, using these data also, we can use this repurposing. Okay, so terbutaline, it is an anti-asthmatic drug. Now also this terbutaline is also used for a neurodegenerative disorder, amyotrophic lateral sclerosis, based on this electronic health records. So these are the 5 to 6 different techniques of in silico repurposing. Now coming to the experimental techniques. Okay, so here the binding assays. Okay, so the proteomic techniques such as affinity chromatography and mass spectrometry have been used as approaches to identify binding partners for an increasing number of drugs. So So the second one is under this you have two techniques, that is SETSA technique, cellular thermostability assay. It maps target engagement in cells using biophysical principles like predict thermal stabilization of target proteins by drug-like ligands. Okay, so one such example is imatinib. It is used for gastrointestinal stromal tumors. And another one technique is our phenotype screening. Okay, so high throughput cell-based screening. So here the phenotype screening can identify compounds that show disease-relevant effects in model systems. Okay, how we do the cell in vitro cell culture. Okay, MTT assay or cell cycle analysis. Likewise, we can go for the phenotypic screening, which relevant to the disease model. Yeah, and this is very important, integrating computational tools. Okay, so none of these methods alone will be sufficiently able to disclose or even model the complex interplay between drugs, target, and disease. Okay, by doing any one method alone, it won't give you the proper what I can say, proper judgment. So always we try to incorporate or integrate one or two tools. For example, if you wanted to find out some a new disease also, new and drug also you wanted to find out, even you can start with the network analysis. Then you can do some structure-based in silico studies. Followed by that, some experimental technique. Okay, so like that we wanted to integrate one or two models. Okay, so the possibility of using one or more computational approaches to navigate through the wealth of available information and hopefully find clues solid enough to justify a repurposing hypothesis worth of experimental investigation. So we have seen already very clearly the step one is hypothesis generation. Okay, so once you generated your own hypothesis, you can properly you can interrogate or justify that hypothesis with a preliminary in silico technique and followed by experimental investigation. Of course, without experimental investigation, it cannot be acceptable. Okay, so the choice of the appropriate methods will basically depend on the nature of the problem. Okay, which problem you wanted to solve, on the type, quality and quantity of information available on the problem in the literature or in public or proprietary databases, because we rely or we depend on the databases which may be publicly available also and some proprietary databases also. So again, the selection of the method, it depends upon the problem what you wanted to solve it. So the message over here is you have to integrate one or more computational tools. Okay, so this is a very effective database, drug repurposing online database. Okay, so here just you wanted to give input, search any drug. It will give you original indication as well as new indication. But again, all these things are a hypothetical level only, a preliminary level. Again, with this lead, you have to do some in silico studies followed by experimental study. Okay, and these are various databases where you can get the details. If you want to have any chemical structure data, you can go for this PubChem. If you want to get any drugs, you can go for this drug bank and therapeutic target database. These are all the databases which will give you the information about the small chemicals as well as the drugs. If you wanted to know the structures of the targets or proteins, you can go ahead with PDB, Protein Data Bank, OCA, okay? And both drug target information, again, you can go ahead with these databases, okay? So these are the databases which are used for the drug repurposing. Of course, we have some limitations also in this drug repurposing. The pathways and mechanisms of many complex diseases remain incomplete because efforts for drug repurposing often require analyzing complete data. Okay. The second is very important. The public databases have increasingly provided enormous information that can facilitate drug repurposing, but yet they are non-synchronized. Okay, because the informations will be updated, okay, monthly basis or weekly basis. Sometimes it may give false information also. And there is no systematic mechanism to obtain fundings to support drug repurposing projects in comparison to de novo drug discovery projects. The ratio is very less. Of course, they have, sometimes the call for will be there, okay, but compared to the basic drug discovery, the drug repurposing project, the funding system from the government, it is very less. And we do have some patent issues, market exclusivity issues, and new method of use, MOU patent can be obtained for a new purposed use of an old generic drug. The patent issues are also there, and we do have some regulatory challenges. So these are the limitations of this drug repurposing. Okay. So with this, we will be moving to a case study, an in silico drug repositioning approach of identification of drug candidates for breast cancer therapy. Okay. So, Dr. Shyam, it's going fine?
Yes, madam, it is going fine.
Yeah. Okay. Is there any doubts now, or shall I continue? At the end of the session, we can take the questions.
Yes, madam, please continue.
Yeah. Yeah. So till now we have seen the proper definition and what are all the steps involved in drug repurposing, the different types, okay, of in silico approaches as well as the experimental approaches and limitations, all those things. So this study, a small study where we have incorporated 2 to 3 techniques of these, what I have explained my previous slides, no, and some experimental study. So this particular study has been carried out by one of my scholar, Dr. Nagaraj. Okay, he has taken this, carried out this particular study. The goal of this research is to identify the drugs for the suitable targets of breast cancer pathways through in silico drug repurposing approach. So here we collect the information of drugs, targets, proteins, genes, and pathways. Then we have constructed the network. 2 types of network we have constructed, protein network and protein drug network. After that, we have carried out the structure-based in silico study. And third study is experimental study. What and all we have proven through these in silico techniques, we real life what happened, that also we have taken. So this is our aim of the study. This research aims to identify the drugs for the suitable targets of breast cancer pathways through an in silico drug repurposing approach. So under this aim, we have 3 objectives. The first objective is to identify the suitable targets for breast cancer therapy and suitable drugs for selected targets through molecular networking studies. That is our first objective. And the second objective is to select suitable drugs for the selected targets by combining structure-based molecular docking and molecular dynamic simulation. The third is to evaluate selected drugs for their target-specific anti-breast cancer activity using suitable in vitro methods. So the first 2 techniques, objective, we have utilized the in silico techniques. Okay. The third one, the experimental techniques, we have used it. So these are the different databases we have explored. Okay, starting with the protein data bank, then top gene analysis, UniProt, GeneCards, Reactome. These are all the different databases we explore. And the softwares, what and all softwares we have used: Cytoscape, CytoHubba, MCode. So Cytoscape is for networking. These are the plugins which are used for along with Cytoscape. and molecular viewer. These are all the Desmond, of course, from Schrodinger we have utilized for the molecular dynamic study. So these are all the softwares and databases we have used. So starting with the first objective, to identify the suitable targets for breast cancer therapy and suitable drugs for selected drugs through molecular networking studies. Okay. So it has been divided. First, we selected the target, then we constructed the network. It is called as protein-protein interaction network. Then the constructed network, we have made some clusters, some groupings we have done. And after that, we did the protein-drug interaction network. Then from this, we selected the drugs. Okay. First, the selection of breast cancer pathway. So these are all the different pathways associated with the breast cancer. So although estrogen receptor ER pathway is the primary pathway altered in breast cancer, it is now well accepted that many other signaling pathways are also involved in the pathogenesis of breast cancer. So among these pathways, we have stick to this EGFR overexpression signaling pathway. Okay, so now this EGFR overexpression, so this pathway and what are all the genes associated with this pathway, we are going to construct the network. That is our first and foremost objective. Okay, yeah. So we have collected using gene card and reactome the genes. These are all the different genes which are associated with this EGFR pathway. Okay. It goes like this. Continue, the table continue. Now, I have so many genes, okay? So from this genes, okay, so it has been narrowed down to 50, okay? How? By using this top gene suite analysis. The EGFR pathway related genes selected from top gene suite analysis. Again, this is this top gene suite analysis. It is a prioritization and gene enrichment analysis. You can see here, there will be some scores. Okay, average scores. Okay, so here we kept the scores, the minimum score is 0.6. So more than 0.6, what are all the genes? What are all the genes which are having more than 0.6 we have considered. And we have narrowed down to 50. So now this 50 genes have been taken for our further step. Okay. So what we did the protein-protein interaction network, then again cluster analysis, and after you construct the network, you have to characterize the network. That is very important, characterization of the PPI network. Then from that characterization, you can finalize the targets, selection of suitable targets. And from the finalized targets, okay, you can do the protein-drug interaction network. Okay, how you can do? The drugs can be collected from various databases. From the collected databases and from the selected targets, you can do the protein-drug interaction network. Again, you have to characterize the protein-drug interaction network. Then you have to identify the drugs. Okay. Yeah. So this is the protein-protein interaction network. So the PPI network, shortly called as PPI network of the selected genes, was generated using the string protein query of the Cytoscape 3.8.2 software. The genes can be considered probably only when the confidence score is greater than or equal to 0.40. The network was constructed without neglecting any of the input genes. Okay. So we know very well we have shortlisted 50 genes. With this 50 genes, we have constructed the network. So this is the typical, what I can say, display of the network. The round, the sphere is different genes. We call it as nodes. And the lines, you can see here, there are various interconnections between these two genes that are called as what? Nodes. Okay, so this is called as node and this is called as edge. Edge is nothing but these lines. Okay, so the network can be analyzed. Once the network has been constructed, it is analyzed using a plugin which is called as CytoHubba to analyze the network tool. Okay, and there are three parameters, important parameters like degree, closeness centrality, and betweenness centrality. Okay, so these three parameters are important, like our how I have given the example, you know, scores. When we do the docking, we just see the scores. When we do the dynamics, we just see the flexibility. Okay, and we should be within that particular RMSD value. Likewise, these are all the parameters: degree, closeness, and betweenness. And once I constructed the network, again I have cluster analysis. I have made that network into two groups by using another plugin which is called as mcode. Okay, mcode app, it is used to create or prepare the clusters, okay, from the parent network based on the functional similarities. So you can see here the cluster 1 has got many genes, many nodes and many edges, whereas cluster 2 is having limited nodes and limited edges. Okay, why? Because the cluster 1 is associated with cancer and specifically related to the breast cancer pathway. Cluster 2 are associated with other than breast cancer, like liver cirrhosis, diabetes mellitus, and cardiac disease. So now this is my area of interest. So I have confined to this cluster 1. Okay. After this cluster 1, I have characterized the topological parameters like degree, betweenness centrality, and closeness centrality. Okay, you can see here the cluster 1 has got 24 genes and cluster 2 has got 7 genes. Okay, so these are all the different IDs of the particular nodes, and these are the degree. So degree is nothing but how each node, that round node, it can make how many edges. So the more degree, the more connections it can make, means it is having more, it has got more control over the network. Likewise, betweenness centrality, closeness centrality means how quickly it can connect on edge node, it can connect both nodes. So these are the three parameters: degree, betweenness centrality, and it is given in the number. So cluster 1, now you can see here, these are the 4 genes which comes top prior. Okay, EGFR is nothing but your ERBB1, ERBB2, ERBB3, ERBB4. These are the top 4 genes have been identified by PPI network. Now, what is my second step? I wanted to do the protein-drug interaction network. So for that, I have to collect the drugs from various databases, starting with Drug Bank, PubChem, and Zinc15 database. So almost 2,637 drugs were collected. And the collected drugs have been again grouped into 4 categories. The drugs indicated for breast cancer therapy, drugs indicated for any cancer therapy other than breast cancer, drugs indicated for non-cancer related disorders, drugs contraindicated for breast cancer patients. Among these 4, our area of interest is drugs indicated for non-cancer related disorders. Okay, so this drugs we have categorized, we have segregated from this 2,637 drugs. Okay, yeah. And from the selected drugs, no, the third category of the drugs have been networked with the selected 4 genes. Okay. The first PDA network of ERBB2. This is the first network. You can observe here degree, betweenness centrality, and closeness centrality. Likewise, EGFR, ERBB1, we have constructed the network. Then we have constructed ERBB4, then ERBB3. Okay, so 4 PDI networks have been constructed. And after this, what we did, the first objective is over. The second objective, to select suitable drugs for the selected targets by combining structure-based molecular docking and molecular dynamic simulation approach. For this, we did the molecular docking studies and followed by binding free energy calculation, then molecular dynamic simulation, and MMPBSA binding free energy calculation. Yeah. So the first one is selection of 3D protein structures for all 4 targets: ERBB1, 2, 3, and 4. So we identified the three-dimensional protein structures. The structures have been collected using UniProt KB and gene cards. After that, using these tools, the various physicochemical parameters have been calculated. Then by using CAPS flex tool, the RMSF value, okay, of proteins have been constructed. Then by protein data bank, the binding sites have been identified. Then the selected proteins with relevant binding packets used for docking studies. These are the description, okay, EGFR, ERBB2, 3 and 4, and this is the PDB ID. And these parameters are various physicochemical parameters, and we are very particular about the amino acids which are present in the binding site of the particular protein. And we did the docking, okay. You can see here all the drugs, okay. Suppose this is the EGFR, ERBB1. What is the co-crystal present in that particular PDB? So that also has been taken. It is very important whenever you do the in silico, that is molecular docking studies, always advisable to take the co-crystal ligand and compare the binding affinity. And the neratinib, the standard drug, which is used for the in vitro studies also has been compared here. Okay, and the calcitriol, the drug which shown very good binding affinity against all the proteins. Okay, so from the various drugs we have taken this calcitriol for our further study. And obviously not only the binding affinity, the scores, we always try to see the various interactions also. What are all the hydrogen bond interactions, particularly van der Waals interactions, alkyl interactions, pi-alkyl interactions. So that also always it should be compared with the co-crystal. Okay, co-crystal and standard also. Here also we have compared. Then this is also very important. We always try to find out the binding free energy, okay, to ascertain the binding stability of the complex. So here there are so many binding energies are involved, starting with electrostatic energy, Coulomb energy, van der Waals energy, hydrogen bond, covalent and lipophilic. Okay, so this binding energy also you can see here compared to the standard neratinib, the calcitriol is showing almost it is similar to that binding energy. Okay. And among these various binding free energies, the Coulombic energy, it shows the major contributor to the free binding energy. Then we have to do the dynamics because it completely, it mimics how the drug acts inside our body. Okay, so the calcitriol complex we have converged with 1.479 angstrom lower average RMSD value rather than the standard. Okay, the stability is more than the standard. Then we have carried out the MMPBSA binding free energy also, okay, to ascertain the stability. Okay, stability of the complex compared to the standard. Okay, want of time, I will be moving very fast. Okay, so the second objective is over. We have done with the selected genes and selected drug. Okay, structure based in silico studies. Of course, I told very clearly you have to prove your hypothesis with experimental techniques. So to evaluate selected drugs for their target specific anti-breast cancer activity using suitable in vitro methods, starting with MTT assay, apoptotic signaling to check the apoptotic activity of the drug. Then to check the cell proliferating, we have done the scratch wound assay, and cell cycle, which phase it is showing the activity, and finally the gene expression study. So the first one is in vitro cytotoxicity screening by MTT assay. So all the studies we have compared with the standard neratinib. Okay, so you can see here the MTT cell lines have been taken. So the IC50 value, when you check with the MCF cell line, the calcitriol shows 44.35, whereas the neratinib shows 35.31. It is more or less similar to that calcitriol's value. So this results demonstrated that calcitriol suggestively down-regulated the MCF7 cell viability, whereas in the Vero cell lines, normal cell lines, they are not toxic. That means they are safe towards the normal cell lines. Okay. So it this data suggested that calcitriol possesses an anti-cancer role in breast cancer cell explosions without causing any harm to the normal cells. And in the second study, we have carried out the apoptotic signaling pathway. So by using various staining methods, so starting with acridine orange Ethereum bromide, so it induces the apoptosis in breast cancer cells as evidenced in the presence of the orange stained cells. Likewise, we have seen with the HOE, CHST staining method, then rhodamine 123 staining. Then the third study is just we wanted to check the cell migration ability. Okay. Whether the drug is inhibiting the proliferating effect of the cancer cells or not. So that we have carried out this scratch wound healing assay. So here the wound is physically made. You can see here. Then afterwards, the drug has been administered, the calcitriol, the test drug, and the standard drug has also been administered. Then at the different time intervals, starting after a day and after 2 days, the wound width has been measured. Okay, you can see here in control, the wound is completely closed. Okay, whereas in calcitriol, the wound width, that is, it has got the anti-proliferating effect. The wound is maintaining, the width is maintaining after 48 hours also. Okay, so this is for anti-proliferating effect. So the cells that were left untreated have proliferated. That is controlled completely. It is proliferated. So the wound is closed and the scratch disappeared at 48 hours. Whereas in the case of calcitriol treated and neratinib treated, a significant difference in the wound healing was observed at 48 hours. And we have gone for cell cycle analysis, in which cycle, which phase, it is very important. So here we have observed different phases, S phase, M phase, G0 and G1 phase. So our calcitriol, it has been acting on these phases, G0 and G1 phase with 74.5% of inhibition when compared to the standard 65.6. And finally, gene expression study. So ERBB2 gene, how the drug, whether the drug is down regulating the particular gene or not. Okay. So there are some standard protocols like RNA extraction, cDNA synthesis. Okay. So, and finally, this is the final results. Okay. So the ERBB2 compared to control, the calcitriol is down regulating the ERBB2 gene. So this is the pictorial representation. So now coming to the summary. So we have carried out 3 types of in silico, 2 types of in silico repurposing and 1 experimental repurposing. So the present study implies a systematic drug repurposing approach for breast cancer therapy. So for what we started, first we have confined to the EGFR overexpression signaling pathway, and we have collected 700 genes associated with this EGFR. From the 700 genes, we have prioritized 50 genes. Okay. And the 50 genes have been constructed PPA network. From the PPA network, we selected these 4 genes. Okay, ERBB2, ERBB1 or EGFR, BB4 and BB3. And the selected genes are again gone for the network construction with the selected drugs. That is 61, 78, 15 and 19 drugs were identified against these 4 receptors. And we have chosen the non-cancer related diseases or disorders Again, these drugs were docked into the corresponding proteins to predict the potential candidates for breast cancer therapy. From these, we found out the calcitriol after our molecular docking study. Then we further validated with molecular dynamic simulation. And these particular asparagine 863, it has got significant hydrogen bonding interaction throughout the simulation. Finally validated with MMPB SA study also to check the stability of the complex. Then further, calcitriol has been checked for various in vitro studies like significant cytotoxicity and apoptosis study, scratch wound assay, cell cycle arrest, and gene expression study. And finally, we are concluding the calcitriol has got significant effect anti-tumoral drug in the treatment of ERBB2 positive breast cancer patients. Okay, and however, it has to be carried out for preclinical study. Okay, so our systematic study explains the calcitriol may possess the anti-breast cancer effect through the inhibition of ERBB2 receptor. So it can be further explored clinically for breast cancer therapy, specifically ERBB2 positive breast cancer. This is our conclusion at the end of the systematic drug repurposing study. Yeah. So these are the 3 publications related to this study. First, we have properly reviewed the network-based drug repurposing and all our in silico studies have been published in this journal, and the in vitro techniques are published in Anti-Cancer Agency Medicinal Chemistry. As I told you, this particular work has been carried out by Dr. Nagaraj. Now he is manager in IPR section at Simji, Bangalore. So with this, I acknowledge my sincere pranams to His Holiness Swamiji and coming from the institution where the institution is governed by a mutt, basically Jagadguru Shri Shivaratri Mahaswamiji, and my sincere thanks to the leadership of Pro-Chancellor, Sir, Vice Chancellor, and entire leadership of JSS Academy of Higher Education and Research. My sincere thanks to the Principal, JSS College of Pharmacy, Ooty, and all my PG and PhD students. And this, I told this work is carried out by one student, and one more student is in front of you, Dr. Shyam Sundar. He has also done the work in this ER positive breast cancer. He extensively worked on BRCA, that is BRCA 1 and 2G. Okay, and I have published the book also. This book is mainly for final year B Pharm students and first time Pharm students with my friend Dr. T. Prabha. So with this, once again, I thank the principal and management of Vasantidevi Patil Institute of Pharmacy, all the faculty members, and those who have joined online. Thank you so much, and I'm happy to answer your queries. Thank you.
Thank you, madam. Thank you very much for accepting our request. And dear participants, you can ask any questions. And before to that, madam, you have explained very nicely about drug repurposing and advantages with
yeah. How the cluster, how the cluster can be prepared. Yes. So again, you can use that M-code app. One plugin will be there. So by using that, you can make the clusters. M-code app.
Yes. One participant asking that, can I get the recordings of today's talk? Obviously, Charlata Namade, we are providing the session in YouTube. So you can, within 24 hours, you can watch there. Madam, today's session, like you have explained about drug repurposing advantages with examples of minoxidil, sildenafil, and thalidomide, successful stories of about repurposing, steps used in drug repurposing, types of drug repurposing, and the tools which are using for drug repurposing like NCBI gene expression omnibus and genome-wide association studies, pathways or network mapping, experimental techniques about drug repurposing, integrating computational tools, drug repurposing online, like various databases of drug repurposing, limitations of drug repurposing, and with the case studies. That case studies is very impressive, madam, and it is very informative and useful to the researchers in the field of drug repurposing, and the publications also very nice. I hope the participants are got good information about this session, from this session. Madam, one the case studies you have follow now, madam. The process it is like a network pharmacology.
Yes, yeah. Starts with
Yeah.
network pharmacology. Actually, first we constructed both networks, PPA network and PDA network. From that, we identified the leads. And finally, and further, we carried out for a normal structure-based in silico studies.
Yes, madam.
Yeah, we did the network pharmacology. Yes.
Yes, madam. Yes, madam. So, any participants you can ask, interact with the madam.
Yeah, we can design some projects also for final year B Pharm and second year M Pharm students also based on these in silico techniques. If you have even online tools only, you can use it. It is like nowadays you can use all the online tools for prediction of ADME studies and even molecular docking studies also. Small projects you can give it for your final year students. Likewise, you can plan for your research. Yes.
Yes. Madam, with your permission, shall I share the PPT screenshots regarding the tools which are provided? Madam,
Definitely. Yeah.
Okay, thank you.
Yeah. And the papers also, I will share you both papers so that
Yes,
if they want, they can use it. That also.
yes, madam. Thank you.
Yeah. Thank you. Thank you so much, Shyam.
So drug repurposing main limitation is that, madam, regarding the patent we can't get, no, madam.
Yeah. We cannot go for patent, and what is my suggestion is even you can go for some analog design of that selected drug moiety, and afterwards you can do patent. Yes. And it will be considered as new molecule. Yes. You can go for sci-fi novelty check, and afterwards you can go for that. Yes.
With the small modification in the structure, we can go for the patent.
Yeah. Of course.
For that, again, we have to go for the preclinical studies.
Yeah, but for that again, you have to go for safety checking. Everything you have to do it. Yes, of course. It will be considered as a small molecule discovery only.
Yes, madam. Okay.
Yeah.
Okay, madam. With this, we are concluding, madam. Thank you very much, madam. And dear participant, we are very thankful to you and your positive feedback regarding last session and today's session. Thank you very much, one and all. And we are expecting the same for following sessions also. Thank you very much, madam, accepting. And it is the words are very small to say thank you. And
It's my pleasure. Yeah.
I'm always throughout the life thankful to you, madam, and you are my part of my family also. Thank you very much, madam.
Thank you,
We are always proud to be a student of you. Thank you very much. You can leave, madam, now.